Joining us today is Dr. Chad Mirkin from Northwestern University. Dr. Mirkin, thank you for being with us.
Great to be here.
You're also the 2015 MRS Fall Meeting Plenary Speaker, and your address titled Programmable Materials and the Nature of the DNA Bond. So tell us a little bit about the themes in your talk.
Well, they're very simple. We're trying to create a new type of chemistry and material science, trying to learn how to break matter down into building blocks called nanoparticles. We think of them as atoms, and then we functionalize them with strands of DNA. So we modify the surface with strands of DNA that become programmable bonds. And then we've tried to develop the design rules for taking different types of programmable atom equivalents, which consist of nanoparticles in DNA, and assemble them into discrete architectures, macroscopic materials, where we can control the placement of all the particles within the material with nanometer precision. And what that gives you then is total control over the properties of the resulting materials as well.
Now, looking through your biography, you've developed a number of lithography methodologies. What are some of those, first of all?
We've developed a whole suite of lithographic tools, ranging from dip pen nanolithography, which is a tool that allows you to use a scanning probe microscope tip to control where chemical reactions occur on surfaces. You can literally draw out structures, small collections of molecules at a time, and generate architectures that have interesting properties by virtue of that control. But we've also created very high throughput systems like polymer pen lithography and beam pen lithography, which get rid of one of the big problems in scanning probe lithography. We eliminate the need to use a cantilever, and we build large arrays of tips that consist of compressible materials that can be mounted in a type of scanning probe tool and then used to deposit in mass molecules from each of the tips over large areas. And we now have arrays with as many as 11 million tips and 22,000 independently addressable tips, so that you have little fingers that can build structures rapidly over large areas made of almost anything.
Now, you're well known for your discovery and development of SNAs, spherical nucleic acids. So tell us about that.
Well, that's actually a really important concept in science in general. It's a very simple one. The most important molecule ever discovered by scientists is arguably DNA. The most important structure ever determined is the duplex, the double helix that defines DNA. The understanding of that structure led to an incredible number of advances throughout biology, medicine, and now even material science. We've learned how to take nanoparticles and arrange those same types of molecules into a spherical form. We can use the particle as a template and arrange the oligonucleotides through chemistry on the surface of the particle into a spherical form. And what's interesting about that is those spherical structures have properties that are completely different from the conventional linear nucleic acids. And those differences, including the ability to rapidly enter cells where linear nucleic acids don't, have led to a huge number of advances in both biology and medicine. And they lay the groundwork for creating a lot of new medical diagnostic tools and a lot of new therapeutics based upon those novel materials.
Fantastic. Dr. Chad Mirkin, Northwestern University. Thanks for your time. Enjoy the meeting.